Hello, today my topic is phylogenetic data. So what does phylogenetic means? Philo in Greek word means group or kind, and genetic is genetic. So also it is about evolutionary biology. In biology, it is the study of the evolutionary history and the relationship among individuals or group of organisms. Use the noun phylogeny to describe the branch of biology that focuses on evolution and the differences between species. Next, we will talk about an example. So before we talk about an example, I want to establish that phylogenetic tree is just basically phylogenetic data. So I screenshot this page from NCBI, and yeah, so more accurately, phylogenetic tree and the relationship between phylogenetic tree and phylogenetic data is that we use phylogenetic data to build phylogenetic trees, and I will talk about what is phylogenetic tree in a second. And after I give you in simple example, I will go back to this page from NCBI and tell you what kind of phylogenetic data are out there. So, as I already say, that phylogenetic is about relationship between individuals or group of organisms, or types of organisms. And as we can see here, there are 5 organisms, or 5, let's say, just animals, and there's lamprey, antelope, sea bass, bald eagle, alligator. And we can see that there are also 5 features. In the simple example, just for easy understanding, we use the features that everybody knows, like feathers, fur, lawn, gizzard, and jaws. So I borrowed this image from a video from Khan Academy in YouTube, and I went back and checked, and the correct word is say that there are 5 species in this page, and there are 5 traits, just for correction. So let's take a look, and we can see that lamprey does not share any of these 5 traits with the other 4 species. So intuitively, we think that lamprey is the most distant species, from the other 4, or among the other 4. And is that really 100% correct? I don't know, but this simple example is just trying to give you an idea of what phylogenetic tree is. And because lamprey does not have any of these traits that we listed, we call it a outgroup. Yeah, let's just set the terminology straight. It's an outgroup. So how do we start drawing this phylogenetic tree? First, we assume there's a common ancestor, of course, based on Darwin's theory, and there is a common ancestor among these 5 species, including lamprey and antelope and sea bass, bald eagle, and alligator. So there is a common ancestor way back to time. And then to the present time, from I don't know, millions years ago, from the common ancestor into the present time, there's a lamprey existing, and we wrote lamprey on this left branch. And then we look back to the table and we see, the other 4 species, except lamprey, all share a common trait, jaws. And then we assume that, from the common ancestor, that red dot there, at some point, getting closer to the present time, there's a derived trait. We call it derived trait, and it's jaws, and the jaws trait from that point on just stick around, and the other 4 species all have jaws. So for convenience, since we have already talked about lamprey and jaws, we just cross them off, and then we look at the rest. We can see that for the rest 4 traits, sea bass does not share any of them with does not have any of them, and the other 3 species do have some of them. So it's only reasonable that we assume that after jaws, at some point of the time, that a sea bass was getting derived, and it just keep existing, and it will maybe it evolved into sea bass, and it exists in the present time. So at this point, I do believe we all know what's going to happen next. So how do we put jaws before sea bass? Because except lamprey, all 4 of the species all share a common trait, jaws. And then after sea bass, all 3 species, antelopes, bull eagle, and alligator, they all share the same common trait, lungs. Well, they all live, outside the water, and they breathing airs. So we will see that after sea bass, a trait, lungs, got developed, and exists. So to make this sound more academically correct, so we say that sea bass and other 3 species share a common ancestor, and one of the child species must evolved lungs after a certain time. So one of the main purpose of the phylogenetic tree is that we are trying to find an explanation of all these species and why they're sharing these traits. So I'm not saying 100% that along the sea bass there's no lungs existing along that time, evolved into sea bass. Maybe at some point along that branch, lungs got developed, and then for some reason it just got replaced or disappeared. It's all possible. So what we're trying to do here is that we are trying to find a parsimony. It means we're trying to find a simple as possible explanation to explain this. When someone calls you parsimony, it means you're cheap, but it just actually means simple in this case. We were trying to find the simplest one. So now I'm just gonna jump to the end, and we all know how it will goes. We will find the next trait, and we will find a next branch. So we have we find where eagle is gonna be, and alligator and antelope. So again, this is the parsimony. It does not mean it's true. It just means we are trying to find the simplest explanation, and there are more complex explanations. And a lot of them, or some of them, might be very true. But however, what I'm trying to say is that from this phylogenetic tree, we can easily see what's the common ancestor and how related these species are between each other, and what kind of common trait they're sharing. Now let's go back to the NCBI slides they provided. So there are multiple phylogenetic data types, and by the way, this slide revised on 2002, so it's pretty old. But I believe these are still true. So the simple example, we use a very simple traits like fur and lungs. However, there are various data types that we can use. Morphological data, And that means morphology means the study of the structure. So we have, number of spines, and we have size and shape, bony projection on the femur. I don't even know how to read that. So we also have behavior data, pitch or duration of frog or bird calls, and we also have molecular data, and we have LDH, isoenzyme patterns, and DNA or protein sequences. And of course, we are most familiar with the DNA or protein sequences. And, well, the same for the NCBI, and they say they will focus on phylogenetic methods used in relation to molecular data. So yeah, I do think our lab can contribute to phylogenetic data. So before we just getting to more of molecular phylogenetics, we want to know, well, what is it exactly? And I think reading the brief history of molecular phylogenetics will help us know what exactly it is. So apparently in 1990, something happened in molecular phylogenetics, and Nuttall discovered that apes are closest relative to humans. Do not ask me some of the words in there, I don't know either. And in the more recent time, there's inducing biosis. Apparently, it means that it's attempting to explain the origins of some eukaryotic cells. It's just trying to, explain the origins of some cells. That's basically what's it about. And I do believe that if we want to know exactly what it means, we probably need to read a lot more articles. And it also helped with the study of divergence of phyla and kingdom. If you don't know what that is, it's the taxonomic rank. So there's species, genus, family, order, class, phylum, kingdom, domain. And phyla is the plural of phylum. So what I'm trying to tell you is that molecular phylogenetics is very useful. It's definitely not useless. And another example is in pharmaceutical research. I personally am very interested in pharmaceutical, and I hope it will draw your interest too. And how people use phylogenetic trees in pharmaceutical is that pharmaceutical researchers may use phylogenetic methods to determine which species are most closely related to other medicinal species, thus perhaps sharing their medicinal qualities. So sometimes you can't even believe how much molecular phylogenetics can do, or how much we can do with genetic sequences, genome sequences. And a great example is that Carl Woese, he created a universal tree of life in 1987, and it just tells us a relationship among basically all lives, and that's absolutely amazing. So there are 4 stages of phylogenetic analysis. Because this is the first time I look into this, I'm not sure if this is absolutely correct or this is the only way. I'm pretty sure there can be revolutions and innovations, right? But according to the slide I found online, it says that the first one is selection of sequences for analysis, and then multiple sequence alignment, tree building, and the last one is tree evaluation. And naturally, we need to learn about trees. It is definitely not as simple as the first simple example with alligators and furs. So there are tree terminology, and I'm definitely not going to go through this today, and I don't even know a lot of it. I feel like we will need to systematically study it ourselves. So yeah, according to the slides, there are root and branches, and there are sisters and nodes. Yeah, so it's a lot like the Bayesian network. And there are also timing in the tree. I'm sure we already basically get the whole feeling from the simple example. So construction of phylogenetic trees are based on shared characteristics, and branching represent the timing of divergence, and the length is a number of genetic changes. So, what can we learn from organisms' genome sequences? Organisms' evolutionary history is documented in its genome. So we are looking at this, and I do believe that this is the comparing of nucleic acid to infer relatedness in a valuable tool for tracing organisms' evolutionary history. And gene duplication increases the number of genes in the genome, providing more opportunities for evolutionary changes. And here's another kind of like example how the ancestor species gene developed and changes over time into different species, which is modern species. So, and then how the sequence alignment can provide clues to evolutionary changes by examining the effect of mutation occurring over time in species with a common ancestor. And I do also like that there is a lot of math involving in this. According to this, you can calculate the substitution rate. I'm not sure what K or T is, but it looks very interesting. And oh, hey, it says K is the number of changes, and I believe T is the time, right? And we also need to study a lot of algorithms for tree construction. So here's a simple method that's introduced. It's called UPGMA. I'm not gonna go into the detail of this because this is a very simple example, and I do believe that it can be much more complex. But basically, it just do the clustering to find the two proteins that has the least amount of smallest pairwise distance. Last but not least, here's a small summary of what kind of phylogenetic software are out there. And yeah, so this is basically my today's presentation. This is the first time I've tried iMovie to make a presentation. There's a lot of recording and there's a lot of editing. So my purpose today is, well, I'm really sorry that I was not be able to spend enough time to cover everything that professors say that we need to cover. Basically, what I did today is just trying to briefly introduce to you what exactly this is and what can we do. However, I haven't really say what's the gaps and some other things. But yeah, I think this is a very intriguing and this is very interesting to me. I hope it can really draw your attention. And there's so much more for us to do when right now it's the age of data. And I am a big fan of life and animals, and I think there's so much more left for us to discover in the world of animals and life. Thank you. And yeah, if you have any questions or anything about how to use iMovie, please don't hesitate to ask me.